Every second, millions of cells divide inside your body. Today, we'll reveal how mitosis and meiosis make life possible. Subscribe and let's get started. Every second, millions of cells divide inside your body. But how does one process create growth while another creates life? Mitosis is the process your body uses to grow, repair damaged tissues, and replace old or dying cells throughout your life. During mitosis, one parent cell divides only once, producing two genetically identical daughter cells.
Each daughter cell receives all 46 chromosomes, 23 pairs, making them diploid, just like the original cell. Before mitosis begins, every chromosome copies itself, creating two identical sister chromatids joined together. In prophase, loose DNA condenses into visible chromosomes, while the nuclear envelope starts to disappear. At the same time, spindle fibers grow from opposite poles of the cell, preparing to move the chromosomes. During metaphase, spindle fibers align every chromosome perfectly along the center of the cell. In anaphase, sister chromatids separate and are pulled toward opposite sides of the cell.
During telophase, new nuclei form around each chromosome set. The cell prepares to finish dividing. Finally, cytokinesis splits the cell into two identical diploid daughter cells, completing mitosis. But producing sperm and eggs requires a very different process, called meiosis.
Unlike mitosis, meiosis involves 2 rounds of division and produces 4 genetically unique daughter cells. Each daughter cell contains only 23 chromosomes, making them haploid reproductive cells called gametes.
Just like mitosis, DNA is copied before meiosis begins, but the similarities end there. During Prophase I, homologous chromosomes pair together for the first and only time. They exchange DNA in a process called crossing over, creating new combinations of genes.
Crossing over increases genetic diversity, making every sperm and egg genetically unique.
During Metaphase I, chromosome pairs line up randomly along the middle of the cell. This random arrangement is called independent assortment and creates even more genetic variation.
In Anaphase I, homologous chromosome pairs separate and move toward opposite poles. After Telophase I and cytokinesis, 2 haploid cells are formed, each carrying duplicated chromosomes.
Without copying DNA again, each cell immediately begins Meiosis II. During Metaphase II, chromosomes line up once again in the center of each cell.
In Anaphase II, sister chromatids finally separate and move toward opposite poles. Telophase II completes the second division, producing 4 genetically unique haploid daughter cells. Each gamete contains just 23 chromosomes, ready for fertilization. During fertilization, a sperm and an egg combine to restore the full set of 46 chromosomes.
This single fertilized cell is called a zygote, the very first cell of a new human life. From this one cell, billions of new cells are produced through mitosis, allowing the embryo to grow into a complete human being.
Cells divide through mitosis and meiosis. Mitosis creates two identical cells. Meiosis results in 4 unique cells. This supports growth and reproduction.
If you enjoyed this lesson, don't forget to like, subscribe, and turn on notifications for more exciting biology and biochemistry videos. See you in the next lesson.